what I'd like to talk about today is variation, but variation not between organisms, but within an organism from time to time and place to place. Namely, how it is that some genes or gene activities are turned on some occasions and turned off on other occasions. This is obviously a very important problem to an organism, particularly to somebody like you who's a multicellular organism and has the same DNA instruction set in all of your cells. It's obviously quite important to make sure that the same basic code is doing different things in different cells. It's important also to a bacterium to make sure that it's doing different things at different times depending on its environment. So I'm going to talk about a very particular system today as an illustration of how genes are regulated. But before we do that, let's ask, where are the different places in this picture? DNA goes to DNA, goes to RNA, goes to protein, in which you might, in principle, regulate the activity of a gene. Could you regulate the activity of a gene by actually changing the DNA encoded in the genome? So why not? Because what? Becomes a different gene. Yeah, that's just the definition. And why couldn't the cell just decide that I want this gene now to change in some way? I'm going to, I don't know, I'll alter the DNA sequence in some way. And that'll make the gene work. Could that happen? Is that allowed? Yeah, it turns out to happen. It's not the most common thing, and it's not the thing they'll talk about in the textbooks a lot, but you can actually do regulation. So the levels of regulation are many, and one is actually at the level of DNA rearrangement. As we'll come to later in the course, for example, your immune system creates new functional genes by rearranging locally some pieces of DNA. Some bacteria, particularly infectious organisms, control whether genes are turned on or off by actually going in there and flipping around a piece of DNA in their chromosome. And that's how they turn the gene on or off, is they actually go in and change the genome. There's some protein that actually flips the orientation of a segment of DNA. Now, these are a little funky, and we're not going to talk a lot about them, but you should know almost anything that can happen does happen and gets exploited in different ways by organisms. So DNA rearrangement certainly happens. It's rare, but it's always cool when it happens, so it's fun to look at. And something like the immune system can't be dismissed as simply an oddity. That's an incredibly important thing. The most common form is at the level of transcriptional regulation, where whether or not a transcript gets made is how it's processed can be different. First off, the initiation of transcription. That RNA polymerase should happen to sit down at this gene on this occasion and start transcribing it is a potentially regulatable step that maybe you're only going to turn on the gene for beta globin and alpha globin that together make the two components of hemoglobin. And you're only going to turn them on in red blood cells or red blood cell precursors. And that can be done at the level of whether or not you make the message in the first place. That's one place it can be done. Another place is the splicing choices that you make with respect to your message. You get this thing with a number of different potential exons. And you can regulate how this gene is used by deciding to splice it this way and skip over that exon, perhaps, or not skip over that exon. That alternative splicing is a powerful way to regulate. And then finally, you can also regulate at the level of mRNA stability. Stability means the persistence of the message, the degradation of the message. It could be that in certain cells, the message is protected so that it hangs around longer. And in other cells, perhaps, it's unprotected and it's degraded very rapidly. If it's degraded very rapidly, it doesn't get a chance to make a protein, or maybe it doesn't get to make too many copies of the protein. If it's persistent for a long time, it can make a lot of copies of the protein. All of those things can and do occur. Then, of course, there is the regulation at the level of translation. Translation. If I give you an mRNA, is it automatically going to be translated? Maybe the cell has a way to sequester the RNA, to wrap it up in some way so that it doesn't get to the ribosome under some conditions, and under other conditions, it does get to the ribosome. Or some ways to block in other manners than just sequestering it, but to physically block whether or not this message gets translated. Well, it turns out that there's a tremendous amount of that. It's, again, not the most common, but we're learning, particularly over the last couple of years, that regulation of the translation of an mRNA is important. There are, although I won't talk about them at length, an exciting new set of genes called microRNAs, teeny little RNAs that encode 21, 22 base pair segments that are able to pair with a messenger RNA and interfere in some ways partially with its translatability. And so by the number and the kinds of little microRNAs that are there, organisms can tweak up or down how actively a particular message is being translated. So the ability to regulate translation in a number of different ways is important. And then, of course, there's post-translational control. Once a protein is made, there's post-translational regulation that could happen. It could be that the protein is modified in some way. The protein's, say, completely inactive unless you put a phosphate group on it, and some enzyme comes along and puts a phosphate group on it. Or it's active until you take off inactive until you take off the phosphate group. All sorts of post-translational modifications can occur to proteins after the amino acid chain is made that can affect whether or not the protein is active. Every one of these is potentially a step by which an organism can regulate whether or not you have a certain biochemical activity present in a certain amount at a certain time. And every one of these gets used. This is the thing about coming to a system that has been in the process of evolution for 3.5 billion years, is that even little differences can be fought over as competitive advantages and can be fixed by an organism. So if a tiny little thing began to help the organism slightly, it could reach fixation. And you're coming along to this system which has had about 3.5 billion years of patches to the software code. And you know, it's just got all sorts of layers of regulation piled on top of it. All of these things happen. But what we think is the most important out of this whole collection is this guy. The fundamental place at which you're going to regulate whether or not you have the product of a gene is whether you bother to transcribe its RNA.